My name is Laura Hughes, and today I'm going to tell you about our efforts to build the NIAID data ecosystem, a federated search engine to discover infectious and immune-mediated disease data. So one of the hallmarks, I think, of the COVID-19 pandemic has been the unprecedented sharing of information in near real time, as research groups around the world have publicly made available wide swaths of data. And this has allowed the community at large to rapidly develop new diagnostics, new therapeutics, as well as new vaccines. But despite the benefits of open data, there are a number of challenges with search and discovery, which make it really difficult for a researcher to be able to find information that could potentially help advance their research. And so one of the first problems is that the sheer amount of information is rapidly growing. So within the NIAID data ecosystem, we've harvested data sets from across different repositories, and we've seen a wide expansion of the amount of data available. So in 2012, we have about 12,000 data sets, and a decade later, there's been an over 10-fold expansion of the amount of information available. And so with all that information, there's huge potential to be able to use it, but of course, it also creates more challenges to be able to find exactly what you're looking for. And so this is complicated by the fact that in infectious disease and immune-mediated research, the data sets are really diverse and can cover a whole host of techniques, everything from omics, molecular data, imaging data, clinical and epidemiology, as well as others. And then lastly, the sharing of this data is distributed across a number of different repositories, including generalist repositories, technique-focused repositories like OmicsDI or SRA, as well as specialist ones focused on a particular area of infectious disease. And so within the distribution of all these different sharing mechanisms, they're all unstandardized with how they describe their metadata or data about data sets, which allows researchers to more easily find them. And so as a result of this unstandardization, it becomes increasingly difficult for researchers to simply find what they're looking for. And while I'm highlighting this in the context of infectious disease and immune-mediated data, of course, this is a generalized problem across all of open data and all of biology. And so the vision for the NIAID data ecosystem is, how can we really reduce the friction in helping researchers identify useful data sets? And so when we were building this, we were really inspired by the idea of, can we build like a PubMed for infectious disease data? So right now, if I go into PubMed and I search for something like COVID-19 variants, that one simple search allows me to look across all different journals, find publications which may be relevant to my research, and then links out to the actual journals themselves, so then I can read the information. And so we wanted to think about, can we build a similar platform which, instead of publications, focuses on data? So a simple search allows you to search across these different repositories, find information that may be potentially relevant, and then link out to the original source to access the data and then potentially use it. And so I'm going to present three parts of how we did this. First of all, what our approach was and how we built this. Second of all, a few details about how we actually went about implementing this data search platform. And then lastly, thinking about, how can we expand off of our work and where we're going in the future. So first, our approach to building a dataset search platform was really focused on three principles: making sure that it was a federated search engine, building translators rather than imposing a particular standard, and then lastly, meeting data repositories where they are. So first, in terms of federation. So we want to be able to access metadata from different sources so researchers can find information regardless of where the data is actually stored. And so the benefit of this is that it retains controlled access within the repositories. So all of the data is stored there. If they have particular restrictions in terms of who can view it, that is retained within the repositories themselves. And so I really see this as a mutually beneficial relationship with these repositories, where our role is really just to sort of shine a light on the work that they've already done, increasing the visibility of these existing repositories, data sets, and tools by expanding how easy it is to find them and potentially expanding their user base. Secondly, we wanted to build translators rather than imposing a particular standard. So to be able to search across these different repositories, we need to combine and standardize metadata from these disparate sources to enable searching across them. And so when we were doing this, instead of forcing repositories to comply with our particular standards, we have tried to recycle community standards as much as possible and build translators between them so that we can convert between whatever particular data model they're using into our sort of common vocabulary. And then lastly, as much as possible, we wanted to meet data repositories where they are. So, we're especially conscious of the fact that, budgets are tight, and we don't want to impose new work for people or repositories to change for our particular use case. And I think the benefit of this is that it really embraces the diversity of repositories. It may be that a repository has built particular tooling or particular data models for their use case. And so we really want to embrace that diversity and then build translators between them. And then lastly, I just want to mention that, this vision of a federated search engine, I think is, widely accepted across the community. And there are a number of different projects which have sort of coalesced around this vision. And so in particular, I wanted to highlight, so about a year ago, NIH had a wonderful search workshop where they presented research from actual research users. And so I think the two things that really popped out to me was the fact that many researchers were complaining that search is really a hard problem and really prevents them from doing their work. And this concept of a PubMed for data, I think, has been cycling around the community for a number of years. And so this has led a number of projects to build, similar federated search engines. So there are big, expansive domain agnostic projects such as Google Dataset Search, as well as NLM is building their own version of a data catalog. And then there are also domain-specific projects such as DKNet, Databed, and many others. And I apologize if I haven't highlighted your particular data portals, because there are a number of them that are doing great work. So next, I want to move on to talking a little bit about how we actually created this generalizable approach to creating a federated search engine in the particular context of infectious and immune-mediated disease data. So first, I'm going to talk about how we built community-based schemas to align metadata to. Second, I'm going to briefly explain our process for harvesting and harmonizing metadata. I'll then give you a brief demo of how we expose metadata in our search platform. And then lastly, talk about ongoing efforts to standardize and augment metadata values. So first of all, in order to be able to search across different repositories, we need a common vocabulary to align to so that they're essentially talking in the same language. And so our approach was to create a schema which unified all of these different metadata from different places off of a common standard. And so as our base, we have used the widely adopted schema.org dataset standard, which has also been developed and extended by the bioschemas community. And so the first thing that we did was we built a minimalistic data set schema, which had the goal of being able to define a small amount of data that a researcher would need to collect in order to capture the essence of it. And so we recycled 13 properties from schema.org and added in 2 specific ones for infectious and immune-mediated diseases, which allowed us to catalog over 300 different data sets in a short period of time. We then expanded the schema to be more inclusive of all the metadata that we would be harvesting from about 15 different data repositories for our NIAID data ecosystem search portal schema. And so this ends up being about 46 different properties, most of them recycled from schema.org, with a few specific ones that we've added in for our own use case. We then use schemas, the schema that we've developed to verify the data, the metadata that we've collected from different repositories. And so to collect this metadata, we harvest it using any number of methods. Basically, any way that the data is made available by the provider, we'll take it and accept it and harvest it in a Dockerized container, which accesses it either using an API, using HTML-based scraping, data dumps, or embedded metadata. This then goes into a custom Python script, which is this translator script that we've built for each of them to take the data properties in the data model given by the repository and translate it to our unified schema. We then pipe this into my colleague Chunli Wu's project, the BioThings SDK, to create an API to access this information. And this powers our front end built off of React, which allows users to search and query it. And then lastly, I want to mention too that as part of this front end, this search portal, we actually also expose the structured metadata that we have harmonized and standardized to Google Dataset Search and other search engine crawlers, so that the metadata that we've compiled is available not only in our own portal, but also in Google Dataset Search. And so the end result of this is that we are able to create a unified search platform from 15 different sources, which at this time composes about 2.8 million data sets. And this includes data from domain-specific repositories, such as the ones listed here, ones that are more focused on particular genomics techniques, as well as generalist data repositories that are part of the GRAY initiative. And you can actually go and access this right now at data.nia.nih.gov. And so we encourage you to try it out and give us feedback on what we could improve on. And so I wanted to give you a very brief demonstration of what this actually looks like to be able to search across different data set repositories. So within the website, there's a simple search platform. So if, for instance, I wanted to go back and search for COVID-19 variant data, I initially can search that. I'm searching across the 15 different repositories and get over 500 different results. I can then filter it down if I'm interested in particular from data from the most recent year. I can see search results, which explain all the metadata, and then I can go and actually access the metadata, the actual data on the individual data website, in this case, import, and then access it through their own access protocols. And so this really streamlines access by searching across all 15 different repositories, and then allows you to link out to actually go and access the data on the individual website to start with. And so lastly, I wanted to briefly talk about how we can actually improve and augment the metadata provided. And so I think the real limitation of this approach is that we have to work with whatever metadata is provided by the researchers, which unfortunately sometimes is not as complete as we would like. And so I want to give you two brief examples of how we are trying to improve that. And so the first is through linking and standardization. So obviously, one of the most important pieces of metadata associated with a data set is a publication, if available, which allows researchers to understand how the metadata was collected, interpreted, and analyzed. And so using any citations that are provided in a variety of different formats, we actually use the PubMed API, which allows us to collect this information, standardize it into structured fields, and then create continuity between different data sources. And so one important aspect of this is that through the PubMed API, we can also link to funding information if available for that. And so the reason why that's important is that, as there are more and more funding agencies which have mandates to share data, this allows a really easy way to think about compliance. So if, for instance, I wanted to search for a particular grant identifier, I can then easily find all data sets that are publicly available with it. And then secondly, we're thinking a lot about how do we actually improve the metadata inside as well as possible, and thinking about linking to ontologies. And so in particular, we're focusing right now on trying to standardize information about species, infectious agents, and health conditions using the PubTator API and translator KP. And so this has two advantages. The first is that it reduces the number of synonyms down to a common vocabulary. And so if you think about the number of different ways you can represent a mouse, you could have the common name, its scientific name, or its taxonomic ID. And so we standardize all of this information so that you can search for any of those fields and be able to find data sets related to mice. And this also allows ontological searching. So Since this is tied to an actual ontology, it means that, if I'm interested not only in mice, but maybe I'm interested in rodent data in general, I can search for either more generalized or more specific search terms, depending on my interests. And so in sum, the NIAID data ecosystem really improves findability by creating a centralized search platform that streamlines access to data while decentralizing the actual data access and storage and retaining that through the federated approach with the dataset providers themselves. Now, with this project, metadata quality is always going to be the biggest challenge. And of course, like everyone else in the world, we're really interested in thinking about machine learning and natural language processing to clean up this unstructured and unstandardized metadata and improve it. But beyond that, I think we need to think about how do we create better tools to make it easy for researchers to share good data and incentives to reward sharing. It obviously takes a lot of time to share data well. And so, how can we make that process easier? And so, I think one of the potential benefits of projects like the NIAID data ecosystem is by sort of helping increase this sort of incentive loop. And so our untested hypothesis is that by making it easier to find data sets, we make it easier to cite data sets, and that will then increase the incentives for researchers to share data and share better data, which hopefully then can sort of increase the cycle and make more data and more and better metadata widely available. So in the last few minutes, I want to briefly talk about where we're going next with this project and some, I think, opportunities for potential collaborations. And so first, all of our core technology is easily reusable. And so all of it is open source and can be taken by anyone and used for any particular purpose. And so that encompasses a number of dimensions, which I've highlighted here on this very busy slide. And so the first is in terms of developing schemas and sharing them and reusing them. And so I don't have time to talk about it, but my colleague Chun-Li Wu has developed this project called the Data Discovery Engine, which sort of wraps all these tools together to make it really easy to develop schemas, to share them, and to reuse them. And so this is what we use to develop our schemas, which are all publicly available. All of the code to harvest and translate between all of these data sources is available, as well as the BioThings SDK toolkit, which allows you to easily create an API. And I want to mention too that Chun-Li is actually giving a talk at BOSC on Tuesday at 11:50 on this topic. So I encourage you to go and see it and learn more about how to create APIs easily. Lastly, the search interface and web application code is all widely available. And then most importantly, the discovery portal for infectious and immune-mediated diseases is available at data.nia.nih.gov. And so please take a look and tell us what you think. In terms of expanding this to add additional repositories, it's pretty straightforward to do this. And so there are essentially two simple requirements. You have to have some sort of structured metadata, and we have to be able to redistribute it. And then the process that we do to actually integrate an additional repository is really twofold. First, we build a harvester to harvest the metadata. And so this will access the data in whatever form it's given, whether it's a data dump, scraping it, or an API. And then the second stage is to build a translator to standardize the metadata to this common schema that we've developed. And then lastly, I wanted to end by thinking about continuing challenges and opportunities for collaboration. And so I've highlighted four of the things that I've been thinking about over the last few years here. And so the first is, how can we think about crowdsourcing metadata cleanup and augmentation? There are a number of different groups around the world who are doing similar things to try to improve metadata. And how can we really take advantage of this rather than repeating each other's work? Second, I've been really fascinated by this idea of can we build a Google Translate to crosswalk between different schemas? As we're building the standard to translate between different schemas, essentially we're building another standard on top of existing standards. And so I'm really interested in thinking about, instead of developing a new standard, how can we build scripts that allow you to translate so that if I have metadata in the NIAID data ecosystem, can I translate that into a representation in schema.org or DataCite or any other number of schemas? Third, how can we incentivize researchers to share better data and better metadata? And so I think that's a really interesting opportunity for these search portals to really encourage people to make it easy to find data and easier to cite data, and then potentially easier to quantify the impact of shared data sets. And then lastly, how do we promote long-term stability of search portals? And so I think this really comes in twofold. The first is thinking about how do we build in, long-term funding so that, these projects don't die after, the expiration of their funding. And then also, how do we really work between all of these different search portals, which are doing similar work in different domains? And so lastly, I just wanted to acknowledge our wonderful team at Scripps Research of very talented programmers and scientists who have developed this portal. And then lastly, this was a huge project which had lots of input from a whole host of people across NIAID as well as the NIH. And then most importantly, I'd really encourage you to take a look at the NIAID data ecosystem at data.niaid.nih.gov. Thank you.